we're all rooting for a cure for cancer, right? Like, it's such a big research area, tons of studies looking for breakthroughs.
It is.
But have you ever thought about this? Like, sometimes even really promising research is
Oh, yeah.
hard to replicate.
It can be.
And that's what we're going to, like, dive into today.
That's what we're doing. Yeah, and this is really interesting because we're looking at a paper from eLife called Investigating the Replicability of Preclinical Cancer Biology.
Okay.
So basically, it's scientists saying, hey, let's try to redo some big experiments from top cancer studies and see if they hold up.
Right. Yeah, because, like, in science, one study doesn't mean something's for sure true. You want to see if other researchers can get the same results.
Exactly. Yeah, you want that solid evidence.
It's like if I have a cake recipe.
Okay.
If I follow your amazing cake recipe,
Yes.
I should get, like, a delicious cake, too.
Yeah. Yes. That's a great analogy. Replicability is super important in science because it helps us make sure that those findings are reliable and we can build on them. Think about it. If a study says, oh, we found this new drug and it shrinks tumors, but then nobody else can get those same results.
Right.
That's a huge setback. It's wasted time, it's wasted money, and it's potentially false hope for a lot of people.
Yeah, that makes sense. So this eLife paper digs into a project called the Reproducibility Project. Cancer biology. That sounds like a huge project.
Yeah.
They actually tried to replicate experiments from 53 influential cancer research papers.
Yeah, it was massive.
Yeah.
And get this, they were only able to complete 50 out of 193 planned experiments.
Oh,
There's actually a whole other paper just about the challenges they ran into when they were trying to replicate these studies.
50 out of 193?
Yeah.
It's not great.
No, it's not a great average.
Yeah.
But it definitely highlights how hard it can be to actually replicate research. And here's where it gets even more interesting. There isn't just one way to say if a repeated experiment is successful. So they actually used 7 different methods
Okay.
to compare the original results with their replicated experiments.
So there are 7 different ways to decide whether or not it worked.
Yeah, it's not always black and white.
Wow. Okay, my mind is blown.
And this study shows that sometimes even just defining what successful replication means can be tough.
So we've got these scientists, they're trying to redo cancer biology experiments, and they're using 7 different ways to look at those results. Let me guess, things didn't always work out.
Well, let's break it down because some of the results were pretty surprising.
All right, let's unpack this. I'm ready.
First, it's important to remember that in science, we often want to see if something does or doesn't have an effect. So we call those positive results when we see an effect and null results when we don't. And this study looked at both kinds of findings from those original research papers.
Okay, yeah, that makes sense. So like a positive result would be like, this drug shrinks tumors. And then a null result would be like, this drug A doesn't really do anything.
Right.
So what happened when they tried to replicate these different types of findings?
Well, this is where the first big surprise comes in.
Okay.
The repeated experiments often found way smaller effects than the original research.
Oh, wow, really?
Yeah.
So like the original study might have said a drug shrinks tumors by a ton, but when they repeated the experiment, it was just like a tiny bit.
Yeah, it could be a much smaller effect.
So how often did that actually happen?
This wasn't just a one-time thing. This was a trend.
Okay.
The effect sizes in the repeated experiments were a whopping 85% smaller than the originals on average. And 92% of the replicated effect sizes were smaller than the ones in those original papers.
85% smaller.
Yeah.
That's a massive difference. What does that even mean for like all those original findings?
Hold on. It doesn't automatically mean that the original research was wrong,
Right.
but it definitely brings up some really important questions.
Yeah.
And this is why replication is so important. It helps us figure out what's really going on and get a more accurate picture.
Okay.
So not a total disaster, but definitely something to think about.
Yeah, for sure. And I have a feeling this is just the beginning.
Yeah, you're right. We still have a lot more to uncover here, especially when we start looking at those 7 different ways of judging the replication.
Okay, yeah, let's get into it. Tell me more about these different methods and how they change the whole picture.
Okay, so those 7 methods for judging a replication, think of them as like different ways of looking at the same data.
Okay.
So some methods are really strict, like they're basically saying the repeat needs to be almost exactly like the original to count as a success.
Oh, wow.
And then others are a bit more flexible. They're like, well, there's going to be some variation when you try to redo an experiment.
Right, yeah, because it's not always going to be exactly the same.
Exactly.
So it's not just like, yep, the repeat worked, or nope, it didn't. There are lots of ways to look at it.
Yeah, there's a whole spectrum. And what's interesting is that the success rate of those replications actually changed based on which method they used. It shows how complicated this is.
Okay, I'm all about keeping things simple.
Right?
Can you maybe break down like a couple of these methods?
Sure.
I want to see how they actually work.
Yeah, definitely. So let's look at a couple of examples. One method looks at statistical significance.
Okay.
So basically, is it likely that we'd see these results just by chance?
Right.
If it's super unlikely, then it's statistically significant.
Right.
Think about flipping a coin and getting heads 10 times in a row.
Yeah.
Possible. Yeah. But
Yeah.
not super likely, right?
Right.
So with this method, a successful replication would have to find that statistically significant result, and it would have to be in the same direction as that original finding.
Gotcha.
But here's the catch. Only 43% of the replications of those positive results were considered statistically significant.
Oh, wow. So that's less than half.
Yeah, less than half. And that shows us that just because a replication doesn't work, it doesn't automatically mean the original research was wrong.
Okay.
It just means we need more research to figure out why those results might be different.
So it's like, that's interesting. Let's take a closer look.
Exactly. Now, another method they used involves what's called a confidence interval.
Okay.
Or it's basically saying, okay, we got this effect size, but it could actually be a bit higher or lower.
Okay.
It's kind of like giving a range of possible values instead of one exact number.
So instead of saying this drug shrinks tumors by 20%, we're saying it could be anywhere between 15% and 25% shrinkage based on what we saw.
Exactly. And this acknowledges that there's always some wiggle room in our measurements.
Right.
But guess what? Only 18% of those original positive effect sizes fell within the confidence interval of the replication.
18%? That's pretty low.
Yeah, it is pretty low.
Wow.
And it kind of makes you think that those original effect sizes, some of them might have been too high.
Okay.
Remember, the repeated experiments generally found smaller effects. So we've got one method that looks at statistical significance and another that uses these confidence intervals. They both seem to be saying that those first exciting findings might not be quite as strong as they first seemed.
Okay, so both of these methods kind of like point to the same thing.
Exactly. And even though these different methods gave different success rates for the replications, they all kind of pointed to the same big idea. The repeats consistently found smaller effect sizes.
So these 7 methods are kind of like puzzle pieces. They each give us a slightly different view, but when we put them all together, we start to see the full picture.
I like that. It's about getting the whole picture.
Yeah.
And in this case, it seems to be saying, be careful about just believing one study, even if it was published in a big, fancy journal.
Right. So science is a process.
Exactly.
It's always testing and refining and building on what we know.
It's a process. It's not like an endpoint.
One study is just a small part of it.
Absolutely. And that's exactly why we do replication. It helps us not get too excited about one breakthrough and focus on building a strong base of evidence.
Okay, so besides these 7 methods, what are some of the big things we can learn from this reproducibility project?
Yeah.
Like, what does this mean for cancer research going forward?
Well, one of the biggest lessons is that we got to be open. If researchers share their methods, their data, even the stuff that was hard, it can help everyone learn and do better science.
That makes sense. Like, the more open and collaborative science is, the better we can all understand these findings and build on each other's work.
Exactly. It's not about saying someone did something wrong. It's about working together to find the truth, to find those better treatments.
Yeah, that makes sense. This whole conversation has really made me think about how we look at science and what we believe.
Yeah.
It's good to be a little skeptical, ask questions, and not just believe everything we hear.
And I think that's great advice. And remember, science is always correcting itself. Being open about our limitations, redoing studies, and having these important conversations, that's how we move forward in a better way.
I think that's a good thing for our listeners to remember.
It is. And before we go on, I'd love to know what you're thinking. What questions do you have so far?
Yeah, it definitely makes you think. I think the biggest thing for me is like, we can't just take one study and believe it, no matter how good it sounds.
Yeah.
We got to like zoom out, look at all the evidence, and think about the limitations too.
Right. It's like reading a headline versus actually reading the whole article.
Yeah.
There's always more to the story.
Speaking of more to the story,
Yeah.
we talk about how animal experiments seem to be harder to replicate.
Yeah.
I'm guessing that adds like a whole other layer to the...
It does. Animal research is so important for figuring out diseases and coming up with treatments,
Right.
but it also has its own challenges.
Right, because a mouse isn't a human.
Exactly.
So just because a drug looks good in animals doesn't mean it'll work the same in people.
Right. And that's another reason why replicating those animal studies and then taking those findings to human research is super important.
Okay, so where do we go from here? We've talked about these challenges, but are there any solutions?
Well, the good news is scientists are working on this. They're trying to make research more open and repeatable. There are some cool things happening, like pre-registering studies.
Pre-registering studies, what is that?
So you basically submit your research plan to a registry before you even start collecting data.
Okay.
That way everyone knows what you're testing,
Yeah.
and it's harder to pick and choose results later on.
So it's like saying, here's my plan, and I promise to share what I find, whether it supports my idea or not.
Exactly. And there's also this movement toward open science. So researchers are encouraged to share their data and methods openly.
It's like making science less secretive and more accessible.
Right.
So other people can look at it closely, try to replicate it, and build on it.
Yeah, exactly. It's about making science more collaborative and trustworthy.
Well, this deep dive's been a lot. Replicability, this reproducibility project, different ways to judge results, animal research, and making science more transparent.
It's been a really cool journey, and I think it shows us how science is always changing and how we're always trying to learn more.
So what's the big takeaway you want our listeners to remember?
Be informed. Don't just accept headlines or single studies as the whole truth.
Yeah.
Look deeper. Ask those questions. Think about the source.
And remember, science is a journey, not a destination.
Yes.
Those setbacks and surprises, they're all part of it. And they often lead to even more interesting discoveries.
Embrace the uncertainty and stay curious.
I love that. Well, that wraps up our deep dive into the replicability of preclinical cancer biology. We hope this gave you a new way to think about scientific research and the search for a cure for cancer.
It's been great exploring this with you.
And to our listener, thanks for joining us on this deep dive. Until next time, stay curious.